Great. All right, for those online, welcome. Glad you could join. My name is Howie C. I'm a product owner at BD Biosciences, and I work in FlowJo product innovation. I'm going to demo a little bit of what I work on before we get started. The product innovation department is who actually make the plugins for FlowJo. So we work with customers. You'll notice that a lot of our plugins have citations. And so these are things that are developed by researchers, and then we work with researchers a lot of times before they've even published, because they'll reach out to us and they'll want to integrate with FlowJo to make sure that what they're creating is usable by outside of their groups. And we create these Java applets that we call plugins. FlowJo is a Java application, and we create this little applet that can talk to R or Python or Java FX and add extra functionality to FlowJo. So if we went out and wanted to add 40 new buttons to FlowJo, it'd be overwhelming. So what you do is you go to the FlowJo Exchange, and that'll be later in the talk how that occurs. But another part of product innovation is making new products, surprisingly. And one of those new products, which will be coming out soon, is called BD Research Cloud. If you have the newest version of FlowJo, without you even knowing it, you are ready to use BD Research Cloud. Those beta test sites are the only ones, such as the University of Florida Cancer Genetics and the ICBR are a beta test site. So this functionality will work if you speak to Andrea. For now, it is just basically a placeholder for when it works for real. But if you download the newest version of FlowJo, you'll notice that there is a BD logo. And if you highlight over it, it says BD Research Cloud. If we hit that button, it will take you to a website that says coming soon. I can hit a special passcode to unleash the login screen so you can actually get to it. Beta testers will have that information as well. But what this functionality does is it creates a cloud environment for features for flow cytometrists and immunologists. So the first thing, the easiest thing, the most straightforward thing would be keeping your FCS files intact with your experiments. So if you use FlowJo long enough, you'll realize that there's lots going on. It is technically, if you use it in a medical setting, it is a calculator, but it's a calculation engine. In the background, it's doing a lot of advanced calculations on tens of thousands, up to millions of events. And the ethics around dealing with raw data is that we never want to alter your FCS file. So we build a WSP workspace file that connects to your FCS files, brings in information, does your calculations, filters your gates, runs dimensionality reduction, and creates this analysis for you. So if all of these are on network drives, USB drive, external hard drives, Dropbox, it's very difficult for FlowJo to keep those intact. So we created a solution that will work 100% of the time for FlowJo called BD Research Cloud. So you have open from BD Research Cloud, save to BD Research Cloud, add files from BD Research Cloud. So FlowJo knows exactly where to find those on our AWS cloud instance. If you actually press this button, it's divided into organizations. So I can actually choose my demo organization here, and I have sub-projects. This is to keep you and your collaborators organized. So I'm working with a group of people. It could be a lab, let's say. It could be a subset of a lab. It could be a group of people working on a specific R01. You put yourselves into a group together, assign yourselves a project called title of R01 here. You can create subfolders or whatever you need to do to keep yourself organized. But when you open up these WSP files, the data is synchronized, everything opens, works. When you're finished, it saves it back up to the cloud, and everything maintains that connection. If I go over, they're going to download and it's going to load it in there and make sure all my analysis is safe. If I head over to that URL, bdresearchcloud.com, you'll notice that there's a FlowJo portal ID. FlowJo portal is our new subscription authentication system. We use the same authentication. So if you're logged into FlowJo, it knows exactly which account to associate it to. I'm going to log in here and then mosey over to the File Explorer. So if you think about it in an actual workflow kind of view, you're going to be sitting at a cytometer. You're going to generate data. You're going to click on that project where you're generating data, and you're going to press the little upload button. If I can find one that not in the right test organization, am I? Yeah, we're in the, I didn't have full permissions in ICBR. So I'm in the wrong org here. So let me switch to one where I do have full privileges, such as the Diabetes Institute. Head back over to File Explorer, click on that project, click on the project files, and then I have this upload button. And now I'll take all those files that I just generated on your favorite BD cytometer or non-BD cytometer, drop them in here, they will upload. I'll demonstrate that quickly by clicking on this, and I have some comp tubes on the desktop, let's say, and I can upload these, and then they will be immediately accessible through FlowJo. So there's no more transporting it somewhere else, getting it to that computer, dropping it into FlowJo, figuring out where to save it, having them disconnected. And you could have it automatically make a FlowJo 10 workspace during this process. If I had yes, it would ask me to name it, and there's more work to be done. Uploads very quickly, and that's going to be available to you. So that's all well and good. Free cloud storage is a great feature. Keeping that connectivity and everything organized is a great feature. But we want to do better than that. We knew we'd have to get traction on this product. So the biggest pain point probably in flow cytometry is panel design. So as a biologist, you're not really trained in physics. Maybe you read a book on it. We have videos, we have the internet, YouTube. But we want biologists doing biology, and we want flow cytometrists focusing on what they do well. So we created, really it was a problem that people have been trying to fix for the last 25, 30 years. But how do you take a biological hypothesis and design a panel for that? For a lot of science, we can just take kits and buy a kit to do that. But in BD Research Cloud, we want to take your inputs, apply that to a cytometer, and then tell you exactly what you need to get great resolution of your data. If you've ever tried to analyze data and you have poor population resolution, you're not getting that separation between the positives and negative, it's because you probably had poor panel design. So we take all of the concepts around panel design, starting with the cytometer. It's very easy to build a configuration in BD Research Cloud. There's actually a file in Diva that you can export and upload, or you can build it manually. Any cytometer this will work for. And then the next step is where we get your biological information. So I'm going to select humans here. I'm a big fan of human research. And I'm going to start adding antigens or population. So a lot of people say, like, what's your panel for T helper cells, right? And they'll tell you CD3, CD4, blah, blah, blah. But really what you're doing is you're transmitting the information of what is biologically a T helper cell. And you can preload those populations into BD Research Cloud, or you can take what we've suggested. So we had some in here like Tregs. I'm going to pop in some CD4 Treg cells and hit add population. And I've added a list of markers that will help me find those. I'm going to add in another population. Let's do naive CD8 T cells. And I can pop those in. And if there's, oops, did I spell naive wrong? Always helps to get the syntax right. Third try. Let's get there. Okay. Naive, we'll do, oh, there's Andrea's custom population hanging out. We'll do naive B cells. And so you'll see what we have now is a list of markers that help us define what these populations are. You could select a clone if you have a specific clone that you have in mind. Most people for extracellular markers don't care. For intracellular markers, massively important, right? So I am not going to have any specifics for that during this this specific biological hypothesis input. So I'm going to hide that and just focus on my definitions here. So my B cells don't express CD3. I'm going to put them as negative. My CD4 cells are high. You can preload these in as well. It's just a BD because we don't know if these are stimulated, unstimulated. Are they thymic? Are they splenic? So we don't try and guess and predispose what those antigen densities are. But if you know that every single time you're getting mouse splenocytes and they are specific antigen densities, you can save these values so you don't have to put them in every single time. So I'm going to punch in a couple for you. Yes, I think experience is better than literature for the simple fact is when you read T cells, they say CD3 T cells. Is that high, medium, or low? Oh, I'll look at the data. What was the brightness of that fluorochrome that they used? If in CD3, obviously we know, but if it's CD394 and it's like, it's in the middle and I'm like, oh, that's FITSy, but did they titrate their antibodies, right? So then that's information that's generally not that detailed in a paper, even though materials and methods are supposed to be to 100% reproduce paper, but that if there's a word limit on it, that is a disingenuous proposition, in my opinion. I should get 40 pages to detail exactly what I did. So experience always better. And you probably are in a lab that does variations on a theme, right? So you have previous data where you can generally go back and look and get some of these questions answered. If you don't know, leave it as a question mark. We know how to answer that. And that means that you don't know what it is. We don't know what it is. We're going to make sure you can resolve it, but also make sure that it's not so bright that it ruins the ability to detect these other targets. So I can go through here and mark several of these. I'm probably just going to delete them for the sake of time. But what we want to do is show the system that there's variability between these populations. And there's a lot of these markers are co-expressed. So on a cell, if I don't co-express CD4 and CD19, I don't care about the spectral overlap that leads to spillover spreading error that causes the loss of resolution that we're avoiding. And if the system knows that, we can push this, and we've tested this with real life experiments, knowing how far we can push the system, how much spillover spreading error is acceptable for tolerance to make sure that you don't totally blow up the experiment. You can't put CD4 on FITC and CD19 on Alexa 448 and expect that to work really, really well. But you can have a lot of spillover and still be able to resolve these things. So I'll do a couple more here. If you want to do some in bulk like CD45, I can just hit high here. We'll give a couple of these. This is going to be low on both, and we're going to call it a day. I'll delete the rest. All right. Actually, Tregs, we really want that FOXP3 on there. And actually, FOXP3, if we can't resolve that, we've ruined the experiment. So what I want to do is mark that one as critical, and that's going to give an extra weighting to that specific marker to make sure that we're going to resolve it. And I'll get rid of IgD and IgM and 138. Okay. So I have 10 floors on an 18-channel machine. That's no longer getting as straightforward once we get into spectral. This is a spectral panel designer if you want it to be. And its heart, it is spectral. But we figured we'd take the hardest use case and work back from there. So traditional cytometers where you're one detector per marker, we handled that just fine. Complete setup. Now we are into where you select fluorochrome. So now you're going to do this laborious thing where you select them all yourself. No, you're going to press a button. You're going to hit generate panels and it's going to do it for you. You've given us all the information that we need, the cytometer configuration and your biological hypothesis now to answer this question. This is an informatics problem. This is building tens of thousands, potentially millions of different panels, scoring them based on very nuanced logic, and it's going to report back the top 10 panels to the interface for you to choose from. So this could take, just to even find the reagent availability for these, could be a weeks-long process, and then actually determining how those are going to perform in your brain and working out which ones to choose is massively labor intensive. We have hundreds of professionals at BD that support panel design and basically have been doing this process for you. Now it's an algorithm, proprietary algorithm that we have at BD. So in a few seconds, it's going to spit back this information back into the interface, and we're going to look at the scores that it's going to provide. Craig. It is BD antibodies and any custom antibodies that you have input into the system. We don't have any contractual relationships with any other companies currently. The BD catalog has over 20,000 antibodies. We have all of the UVs because those are ours, so you can't get them anywhere else anyway. But if you wanted to put in a competitor antibody, and I've actually made it the system where you can, it's like, I want this on all of these markers. You can kind of do it in bulk, and we even have a bulk uploader. We just can't put them in for you right now. But you can get those into the system. We want you to use what's in your fridge. We're not trying to make you buy a brand new panel every time. It's actually the most common use case is I have a 5-color panel and I want to go to 8, or I have an 8-color panel and want to go to 12. How do I best do that? And what you would have done if that demo included, and let's go ahead and do it, but we have these drop downs where you can pre-choose some of your choices, and then we'll fill in the rest with the best logic from that perspective. So we do accommodate that. We give you the results on the common, spectrum viewer. Really spillover spreading error is in the context of the optical configuration of your machine. So this is a representation, but like this isn't the amount of overlap really between these two things. You have to take those filters into account. Actually, those are right next to each other. So that's actually a pretty accurate representation, but it doesn't know your voltages and stuff like that. But we simulate optimal voltages. And so as long as you're taking that into account during your machine setup, then everything's taken care for. So our top panel is 15.54 is its score. Like golf, less spillover spreading error, less loss of resolution is better. 15.54, 15.61, 15.62. We can scroll all the way to the bottom, 15.72. So basically we gave you back 10 panels that are all going to perform almost exactly the same if you wanted to choose based on which ones are already in your fridge, or I hate APC size 7. Whatever those reasons are, you can make those decisions. You can visualize the results. I'm still working on the mathematician that I work with on this project, Peter Mage, who's amazing, but he's displaying the weighted scores here, which I think is disingenuous. So it looks like we've done a terrible job with FoxP3 and providing resolution, but that's with all of the weighting that we applied to penalize a low antigen density target in the context of a critical to resolve antigen. So really it should be displaying these, I think, as a bunch of panels. And you would choose where you could accept the most spread. So you as a user would say, you know what, I don't really care about, I think CD27 is where I can handle a lot of spread, especially on my Treg population. So I'm going to choose this panel for kind of that arbitrary reason. But as a whole, these panels are going to perform about as well. If you want to download a CSV that contains all of these specific scoring on the specific populations, you can hit that download report button, and it should open in a second on my computer, and we'll see those score values. But yeah, if you really wanted to dive into a bunch of numbers here and see what's going on, we give a full panel score and an individual panel score, individual marker score for each population. So we disclose that all to you for your decision-making choices. What we haven't done is convolute the process with reagents. A good panel is how appropriate the fluorochromes are for the target on the populations that you put together. So once you've chosen your fluorochromes, on the next step, you will actually choose your test sizes, your clones. Like I said, you could have restricted the clones option in the beginning. You can also select them here for arbitrary reasons. If you want to select all 25 or 100 test size, that's where you make that decision here. If you're using a lot of brilliant dyes, they are polymer dyes, they bind together. So you want to make sure whether it's our reagents or competitor reagents, if it has that brilliant label, you need brilliant stain buffer, we give that to you. We have a code that I know we provide to get it for free. I think once this launches, we'll just throw it in the box with everything else, so you don't just making it known. The last part of this process, you hit finalize workflow, and we get some new buttons. The first is generated experiment. So if you've ever sat in front of Diva and done that reagent filter mapping, so your cytometer knows where APC, presumably it could know where APC should be detected. It has no idea what reagent you've chosen. So you have to sit in a machine and say, CD3 is on APC, CD4 is on PE. And you have to tell it that. Since we already know what reagents you're using, we can create a Diva experiment where all that's mapped out. We can also make some doublet discrimination and some extra gates to go in during that process. You could start labeling your specimen and tubes here. So when you show up to the machine, instead of sitting there and doing a bunch of data entry for that process, you just show up, your experimental layout is done, all your specimens and tubes are labeled, and you can start recording. And that's our goal. If you like a quote, we know a lot of people build this stuff and it's got to go to purchasing. You could request a quote. You can email this to somebody that actually is in the punch out system and can input all of those things for you. And all of this is saved in the system as a workflow. In BD Research Cloud, we have this concept of workflows. We talked to a lot of core managers and a lot of immunology labs, and they said, you know, we have these protocols that are in Word documents, but we're not really managing the lab in very smart ways for communicating with each other. So we've created template workflows for instrument startups. Every site is different, so it's probably more appropriate for everyone to create their own. But what you want to see as a core manager is that these technicians that you brought in to assist you with their day, and you can see that that process is occurring. If you're an immunologist that's going to show up to a machine at 2 o'clock, you don't want to find out that nobody started it up. You don't want to find out that nobody ran QC. And generally, you can't find that information until you're at the machine. So we're starting to make a cloud service where you log in. And the heart of BD Research Cloud really is the cytometer. So you can come here and see which machines are up and running. You can see their optical configuration. And this is a newer product. We're very aspirational with all the features that we want to include. Directly connecting with the machine is something that I really want to do, and it's really difficult for security reasons. But someday I think we can get there through working with our security departments. I've already had many conversations with the UF security team and figuring out what we're going to need to put a cytometer agent on the computer. And then BD, in the coming years, we'll have new acquisition software that will hopefully facilitate more of these interactions. Like any software that's collaborative, it has groups of people, you have user accounts, you have permissions, there's an activity log. You get your own little space to work in called your sandbox, or we renamed it just your individual org. In software, there's a term called sandbox where you just get to play around with stuff and not have to worry about it. But you get your own space, you get a specific number of gigs, so you can use all of this for free. There will be premium features at some point, but everything I showed today will come with the base, and UF being a beta test lab will probably have a perpetual premium license for as far as I can get away with that. So with that, I'll turn it over to your new application scientist, Veronica. She is a former postdoc at Emory and has come to not only introduce yourself, but also train you on all those extra buttons in FlowJo that you can turn on and add functionality and put some pretty advanced features. So here you go.
Thank you.
You're welcome. Yep, and I should probably stop sharing the screen first. Too far. Or maybe I never shared my screen.
I saw it.
Okay.
Yeah. We do have a question, Howie. Sorry,
Yeah.
we're getting a little feedback here. There was a question in the chat. When will the research cloud be available to us at UF?
It is currently available. You can send a request to me or more appropriately, Andrea, and we can add you to it, and you can use it for panel design on all the machines that we've added, which I don't think is all of the machines yet, but I could work with her and make sure whatever machine that you want to do panel design should be available. Or Patrick, how about we just throw Patrick's name in the hat too, some of the content. So right now, the manual app is like very drag and drop, but what we want is like streaming together as files to the cloud. So I'm trying to work with the legal team to figure out legally how we get scientific data, just general, streaming to clouds. And with DBIVX cartridges, there's not individual logins that we can align with. So there's some specific. But yes, much tougher problem. Yeah, but you have lawyers in the room, so I can't figure it out.
All right, I'll just do a quick sound check if somebody in the virtual audience can tell me if they can hear me and see my screen. That would be great. Okay, great. All right, well, hello, everybody, and welcome back for everyone who was here this morning, either virtually or in person. For those that haven't had the pleasure of meeting me yet, as Howie said, my name is Veronica Obregon-Perko, and I'm a FlowJo application scientist with BD. Been in this role for a few months now, but I'm really enjoying it so far. And this is probably my favorite part of the job right here, is interacting with all of you. So for those of you joining us virtually, please don't hesitate to type your questions in the chat throughout the presentation if you have any questions. And so my plan today was I tried to offer a balance of giving you some information about all of the high dimensional analysis tools that you can use within FlowJo, but also incorporate a little bit of demo. I get little half and half feedback where sometimes people want more slides because they want more information, but then some people get angry that they didn't get enough demo and they don't actually know how to use them. So I've hopefully achieved some kind of a middle of the road balance here. But if you need any information or would like some more guidance, please come talk to me after the talk. And for those of you that weren't here this morning, I have some business cards up front. I've also given a handful to Andrea so she can distribute it to her core users. All right, so let's go ahead and get started. So at least for me, as a measly postdoc, which was only 2-3 months ago, when I'd heard about high dimensional analysis tools, I'd always kind of just thought of them as an added bonus to your high parameter analysis, right? I don't know how many of you are familiar with these as seen on TV ad products, where they basically advertise as products that you could probably live without, but if you bought it, your life might be slightly easier. So, you know, there's a woman here having trouble scooping her ice cream, so they're marketing a heated ice cream scoop. There's a man here who wants to drink water from the sink, but apparently can't do that or get a glass, so it's installing a water fountain for your sink. Got a woman here who can't open a jar, so selling her some kind of gripped jar opener. Perhaps she doesn't have a husband around or a stronger partner to help open the jar. And then we have a man up here who just can't seem to manage his TV dinner, needs some kind of a better stand. So what I'm hoping to do today is do a better job than these as seen on TV ads of trying to convince you that high dimensional tools are not going to just make your analysis a little better for your high parameter panel, but are really going to be a necessary part of your toolkit. And you can spend your time, you know, scooping your ice cream a little harder, but hopefully FlowJo will give you that time back in your analysis. So if that doesn't convince you, let's tell a little story, right? Let's talk about a hypothetical experiment. Hypothetical air quotes, because this is something I did once. Hypothetical experiment. I want to identify immune correlates of viral control in an animal study. Okay, I've designed a high parameter panel, about 26 colors, to phenotype these cells. So here's kind of an idea of the gating strategy. So we've got some markers for major lineages, mainly T cells, and then I've got a slew of activation, exhaustion, and function markers. So let's say I've got about 10 of those. Okay, so I'm told, okay, Veronica, I want to know the expression of these markers on CD4 and CD8 T cells. Okay? So that's 10 gates on each of my CD4 and CD8 T cells. That's not too bad, maybe. But then I'm also told, well, but I want to know the T cell subsets too. So now my gates have kind of logarithmically increased, and my gating strategy is becoming quite complicated. Also, each of these blue boxes represents a single positive population. What if I want to know double positive, intermediate, double negative, triple positive? Right? These are all gates I'll have to make, all statistics I'll have to generate. And so you can see that, you know, you might end up in a situation where your collaborator or your PI is knocking down your door asking for this analysis, and it's going to take a long time, right? You're going to be dodging people in the hallway. Funny story with this picture. I actually took it last night. I was leaving my hotel. Yeah. I was leaving my hotel to go to dinner and was you know, faced with some violence on the streets. Yeah, I was even wearing my chubby mermaid shirt because I had just swam with a manatee, which was super exciting. That's right. I did not disturb the wildlife. Also, I was a little scared of wildlife, as you can tell. But the point I'm trying to make with this photo is you don't want to be dodging people in the hallway, right? You want to get your analysis, you want to run with it, and you want to do it efficiently, effectively, and answer the most questions you can. And we want to give you the tools to do that. So you can see there's 3 main challenges that I'm trying to convey with this very embarrassing picture. One is you're going to have countless manual 1D or 2D gating, as you can see from that tree that I just gave you, which is a very simplified view of what the gating would look like. Also, I might miss intermediate or rare phenotypes, right? So what if I don't know that there's a very small frequency of cells that express, you know, both CD38 and CD69, but they're not T cells or something like that. Or we might miss intermediate expression levels. So a lot of these immune markers, especially chemokine receptors, can be expressed on a gradient. So if you do just a simple bimodal gating, you could miss something very important. And also you have to maybe know what you're looking for if you're going to do manual gating. So what if I don't know what kind of cell types I should be looking for, what kind of markers I should be looking for, what kind of patterns? So there's really a lot of knowledge that would, you know, have to go into an effort for defining your populations manually when you have so many different parameters that you want to look at. So I'm having trouble going to my next slide. There we go. So this is where some computational analysis for high parameter panels might come in handy. So using these tools, the complexity of multiparametric data can really be tackled through some kind of an exploratory approach. And so that comes in 2 main forms. and that are complementary to one another. So we have dimensionality reduction, which will allow for visualization, and clustering, which is going to allow for automated population identification. So you can see that as an example from this publication down here, where they've dimensionally reduced their high parameter data set onto a two-dimensional space and manually gated for a series of T cells and monocytes and other types of cells. But they've also used an automated clustering algorithm to identify populations, and this is completely unsupervised, so it's not telling it what to look for. It's just looking for populations, and we'll talk about how that works. But you can see if you color code them that it's identified 15 populations that all roughly correspond to a population that was found through manual gating. But I can guarantee the bottom graph probably was generated a lot faster than the top graph. So it's just an idea of what you're trying to accomplish with these tools. So ultimately, with using computational analysis, you're going to be able to get a more thorough and unsupervised assessment compared to what you'd be able to accomplish with manual gating. So ultimately, you can call, visualize, and enumerate your phenotypes between conditions, treatment groups, et cetera. Trouble. All right, there we go. So let's just do a bird's eye of the analysis workflow and how that's going to go. But I'm going to dissect each of these a little bit just to give you a little bit more background information. But ultimately, this is what the workflow for high dimensional analysis could look like. You might start with a cleanup gate down to your populations of interest and remove any anomalies, dead cells. And then we're going to normalize the input of all of our samples of interest before we do a concatenation, which is basically a fancy word for merging. We want to merge all the files of interest. So then after we do that and assign keywords to be able to tease them apart later, we can do a dimensionality reduction to visualize the data in a low dimensional space, do clustering to identify populations, and then explore those clusters and ask what those populations are and how do they differ between different treatment groups, different tissues, different donors. Right, so let's go into each of these. Cleanup and concatenation. So cleanup, much like if you were doing a manual analysis, is very important to the data. You want to remove any debris or dead cells that is going to introduce artifacts into the analysis. And you can also remove anomalies using some of our cleanup algorithms. Some of you may have heard of FlowAI or FlowClean. So these are algorithms that can help remove anomalies from signal acquisition or negative or positive outlier events, or some issues with the flow rate. So we do have algorithms to help with that. You can also use a nice time gate to get rid of some of those anomalies. So after doing this, then you'll want to gate down. Oh, question? Yeah, so that's a question in the audience. Is there a minimum number of events? I would say maybe in the thousands, 5 to 10,000. You can technically do it with less, but I think that you're going to find that any maps you generate are just going to look very spread out, a lot of white space. Clustering, you might have clusters that only have like 2 cells in it or something like that. So I think more is better to a degree, but if you're dealing with something like a rare sample, maybe you can try to move on with it. Let's see. Okay, so then gating down to populations of interest. So common question is, what population should you focus on for your downstream analysis, right? Like, can I just do all live lymphocytes or do I have to do T cells, NK cells? And really that's up to the question that you want to ask. If you do a more bulk population, you'll get a more bird's eye view of everything. If you zone in on, say, NK cells or T cells, then you might get a bit more resolution for some of those subsets. So really that's up to the researcher and what they're interested in. Then the next step is you want to add keywords to designate your sample type. So we'll show that in the demo. But ultimately, once you merge the files without keywords, you'll be unable to tell them apart after the concatenation event. So we add keyword metadata so that later after we merge, we can use gating to tease apart, say, different tissue types or different donors. And then when we're merging the files, we also want to normalize the number of events across the files. So you don't want one particular donor dominating the merged file and kind of influencing all of those phenotypes that ultimately get identified through clustering. So you want them to be evenly distributed so they all have an equal chance of contributing to the ultimate profiles that are going to get generated. And then after the merge event, and you could do it before too if you'd like, I also wanted to drop a line about scaling. So when we're doing these dimensionality reduction or clustering tools, it's completely unsupervised. You're not going to be guiding the algorithms with gates that you made. So the algorithm is going to look at your data across the white space, and it's going to decide what it thinks a CD3 T cell is or what it thinks is a CD4 T cell. So think of the algorithm as having eyes with no gates and no scale values. Okay? So you can see in this example, if you improperly scale your data, like as done here on the right, where you've artificially created two CD3 populations, but really this is the true CD3 positive T cell population. This is just an artifact from improper scaling where I've now created two CD3 negative populations. But my algorithm is going to see this and think, oh, well, I have a CD3 negative, a CD3 high intermediate, and then maybe some debris off to the side. So scaling is definitely important. Proper scaling is going to give you more rich clustering and dimensionality reduction. And I pulled this figure from this recent review paper that came out that talks about the pitfalls of high dimensional analysis. I'd highly recommend it. It was a very informative paper. It's about 2-3 pages long, so you can read it over a lunch break, but it talks a little bit more about scaling and gives some good examples of what can go wrong if it's not done properly. So another question we get is, why do we have to concatenate the files in the first place, right? So if I'm merging everything, how am I ultimately going to make comparisons between groups? Well, that's where the keywords come in. But the reason we have to concatenate is because all of these algorithms are stochastic, meaning that they're dynamic and they're not going to make the same result every time that you run the algorithm. So they're completely random. Even if you ran the same t-SNE on the same sample multiple times, you're going to get different looking islands all the time. So the only way you can compare samples within a t-SNE is if they were in the same t-SNE or in the same UMAP. So concatenation is absolutely necessary for any kind of comparisons. You can't compare two different t-SNE trees to one another or two different UMAPs to one another, because they're just, it's not going to be a fair comparison. All right, so let's talk about dimensionality reduction. So now we've cleaned up our files, we've added our keywords, normalized, concatenated, ensured proper scaling. We go into dimensionality reduction, which essentially is what it sounds, provides a low dimensional representation of high dimensional data, such that events with a similar multidimensional expression pattern will end up grouping together on the dimensionally reduced space. So if you imagine just a big bowl of Skittles and you had to lay them out on a table and they're just getting organized, say, by all the citrus colors, all the berry colors. So it's kind of just laying them all out nice and flat, and you can start separating them into islands of similar phenotypes. So that's kind of demonstrated down here in this third parameter heat map. So you can see this dimensionally reduced t-SNE. And if I ask, where is my CD4 marker, I can see this hot spot of yellow where all my CD4, presumably CD4 T cells, have ended up. Same for CD8, CD19, CD56. So some of you may be thinking this sounds reminiscent of PCA or principal component analysis. But one key difference is that most of these dimensionality reduction tools are optimized for nonlinear embedding, as our PCA is going to be better for linear data. So PCA isn't alone entirely suitable for flow cytometry data. Question? Yeah, you could. And we'll show a little bit in the demo of how to, start teasing into what these islands might be. I would say a t-SNE alone would be tricky to use for population identification, but it can definitely get you started. Yeah. No. 2 questions from the audience, 1 planted. What was the question again? Yes, can you backdate on an island to get an assessment of its ancestry? Yes, you can, and we're going to talk about that during the demo. Is it the best way? Probably not. There's some other tools that can help you with population identification, and we're going to get into those. Okay, so that's dimensionality reduction. So what are my options for dimensionality reduction? So I've shown t-SNE a lot as an example, but I do want to highlight that we do have some other tools. So t-SNE is probably the most popular, probably the one you've heard the most or seen at any flow cytometry conference poster. So this is T-distributed stochastic neighbor embedding. So it uses Gaussian probability distribution, looking at nearest neighbors and segregating them into these islands of similar phenotype. So t-SNE is really great for flattening data very well. You really get a nice look at all of the events in a 2D space. You don't really necessarily have big blobs of cells hiding behind other cells. So you really get some nice resolution within the islands. And so that's what we refer to as local structure. Now, let's go on to UMAP. So UMAP, or Uniform Manifold Approximation and Projection. So it is also a dimensionality reduction technique, but it makes its map on a slightly different mathematical algorithm. So it uses topological structures or geometric structures to approximate the way the data looks in the high dimensional space, and then uses those to map it into a two-dimensional space. So it scales very nicely to large data sets. One advantage of UMAP, as well as some other dimensionality reduction tools over t-SNE, is that it does a better job at maintaining global structure. So with local structure, we're considering events within an island. For global structure, we're considering the relationship between islands, right? So with UMAP, you can see that you sometimes get these little offshoots or peninsulas, and those can give you some inference about maybe trajectory or intermediate phenotypes going between islands. And you can see as another example, you have a purple island here and a blue island. So those NK cells and CD8 cells, which we know have some similarities biologically, but we can't make those same conclusions using a t-SNE space. So if you come down here, for instance, you can see my B cells have gotten separated a bit. And also I have monocytes close to B cells, but monocytes are not really like B cells. And so them being in proximity to one another on a t-SNE space doesn't mean anything. So you can learn a lot of resolution information within an island using t-SNE, but you can't make conclusions between islands and their distance to one another. But you can do that using a UMAP. So depending on what kind of question you're asking, they each have their advantage. So t-SNE might be nice for comparisons between treatment groups because you get a nice flat view of everything. As where UMAP, maybe that's going to be better for some kind of a cell trajectory study or a study that includes cell trajectory or monitoring cells over time for after repeated treatments, and you want to see the evolution of a cell phenotype. So they each have their merit. TriMap, relatively new, so it's also similar to UMAP. It preserves global structure very well. So it uses a PCA initialization modified with triplets to optimize the dimensionally reduced space. It's pretty fast, and it's thought to have a higher fidelity for a global structure. So you can see that you get an even clearer picture of some of these peninsulas projecting between islands. So it is a favorite for cell trajectory studies, and it's also becoming very popular for single cell RNA-seq. All right. And then lastly, we also have EmbedSum. I can't say it's my favorite dimensionality reduction tool. It basically is just a dimensionality reduction of one of our clustering algorithms, FlowSum. And so it is pretty fast if you just want a quick visualization of what your clusters look like and how they relate to one another on the dimensionally reduced space. And it's pretty fast for high volume data sets. But as you can see, it's a little fuzzy at times, depending on what the data looks like. So I think it's a quick way if you're more so interested in just getting right into your cluster analysis and you just want a way to visualize it. But I would think the others have a little bit more merit. All right. So we dimensionally reduced. Now we want to identify populations, right? And this is where clustering algorithms come into play. So clustering provides unsupervised identification of populations from the high dimensional space. And so ultimately, phenotypically similar cell populations will end up within the same cluster. And there's going to be, ideally, heterogeneity in expression of markers between clusters. Okay? So this is just a simplified picture from Phenograph, one of the clustering algorithms. And you can see that it has organized all of the events in the high dimensional space into these clusters or communities. And it has this community here based on its relative expression of this marker, CD34. So we have high CD34 here, and then each of the other clusters might have no expression of that marker or high expression of another combination of markers. And so if you zoned in on a particular cluster and looked at its phenotype should be different informatically and biologically, hopefully, from each of the other clusters. And you can also visualize those with an overlay on the dimensionally reduced space. And if you're using something like UMAP, you can maybe make inferences about the distance between one another on the dimensionally reduced space. So clustering options in FlowJo. FlowSum self-organizing map uses k nearest neighbors to generate that. It's fast, very precise, meaning it generates a reproducible knowledge. So knowledge about cell types we already know exists, so it reproduces those cell types very well. Another added bonus is you can apply new data to an existing map. So if you recall, I mentioned you shouldn't be running multiple algorithms for multiple comparisons. You should concatenate everything and concatenate everything and run the same algorithm. But in the case of FlowSum, you do have the opportunity to generate a clustering off of a concatenated file and then apply new data to that clustering map that gets formed. So that is one nice advantage of it. Another advantage is it does generate a visual. After you run the algorithm, as I'll show in the demo, it generates a minimum spanning tree, and you can get some information about each of your clusters and their relative marker expression. So a couple caveats is you do need to specify the cluster number. So you do have to help the algorithm a little bit in terms of finding a good cluster number. And although it reproduces prior knowledge populations very well, it doesn't subset very well. So it doesn't tend to find rare subsets very well, or a subset even particular, like if you had T cells and you were trying to find an activated memory T cell or a non-activated naive T cell, might have a little trouble finding some of those more refined subtypes. But if you're looking for just some automated cluster population for cell types that only you're interested in are the ones that we already know exist, then it's a very nice tool. We also have phenograph, which is probably one of our more popular clustering algorithms. So it uses nearest neighbor graphs to generate communities. And so unlike FlowSum, it does auto pick the cluster number, tends to overcluster, but because it does that, it subsets very well. So a very nice tool for finding more rare populations, more intermediate phenotypes. It has a nice coherence, so it generates a nice pure clusters that are very different from one another. So a couple of drawbacks, it's probably the slowest clustering algorithm, so you have to be a little bit patient, and it doesn't generate any kind of visuals. So these are just thumbnails from the FlowJo exchange. It doesn't actually generate an image to look like this, at least within FlowJo. Lastly, we have XShift, which uses weighted k-nearest neighbor density estimation. Like phenograph, it auto-picks cluster number. It's faster than phenograph, so it's a nice alternative to phenograph if you're just very impatient and you want your clusters right away. And the authors have optimized it to find rare populations. So it, like phenograph, tends to overcluster, tends to find rare populations, and doesn't penalize outlier events, but rather considers them to be rare cell types. Like phenograph, doesn't generate any visuals. So there are a number of clustering tools out there. I just went ahead and covered the three most popular and some of the three of the four we offer within FlowJo. But a lot of the information that I gathered for this presentation has also come from this very nice review paper, or I guess it isn't necessarily a review paper, but a paper that basically puts all of these clustering algorithms head to head. And it generates this decision tree to help you with your data set pick a good clustering algorithm. So care about known cell types only? Well, pass jail, go and go straight to these. You want to find only rough cell types? Maybe you want to use FlowSum. You want to find a large number of clusters? Maybe you want to use XShift. You want to find refined subtypes? Maybe you want XShift or phenograph. So it's a really nice decision tree. I have it printed on my desk and I look at it multiple times a day. So it's a very nice tool. I highly recommend giving the paper a read. And so you can see how some of these clustering algorithms were pitted against one another. So we have coherence, which is again purity of the clusters and their difference from one another, and reproducing prior knowledge, and the number of clusters that get formed. So you can see how FlowSum falls somewhere in the middle, whereas XShift and phenograph tend to generate more clusters, tend to have a little more coherence, but don't necessarily produce prior knowledge. So I was just on a call with a user the other day where they sent me this picture. This isn't the exact picture, but it basically looked like this. And they said, well, I ran the clustering algorithm, but this is all I got. This is all FlowJo made. Like, what am I supposed to do with this? Like, what does that mean? All right, this is just showing you the frequency of your clusters after it finds those populations. So yeah, you could take those cluster populations, overlay them on a dimensionally reduced space, kind of just look at them, see their relationship between one another if you used dimensionality reduction with maintained global structure. But the best way to really identify these phenotypes of these clusters, getting to the question that we had from the audience earlier, is you can explore clusters with, get ready for this, Cluster Explorer. Yeah. So very appropriately named tool. It is one of our, probably my favorite plugin available through FlowJo. That's why I put a little superhero on it to explore the clusters. It doesn't actually look like this in the FlowJo website. But the reason I put it there is this is really going to be your best tool for really diving into what these clusters are. Because if you've chosen a clustering algorithm that isn't going to just reproduce prior knowledge, ultimately you're going to want to know what these clusters are, what cell types are in there, and how are they different from the other ones. So this is just a snapshot of what that plugin looks like when you fire it up. So you get a lot of information. You get the frequency of each of your clusters. You get these chord plots that show the relative expression of each of the markers in your panel across each of the colors. I'm sorry, each of the clusters represented by a different color. You also get a heat map view of that same information. And you can also visualize your clusters within the dimensionally reduced space. And so the great thing about this tool is that it doesn't just generate static images, but it's completely interactive. So you can say zone in on cluster 3, which is pink, and look at its marker expression level and look at it as a heat map view. You can see it on the dimensionally reduced space. This 2D plot, you can change the axes if you just want to see, if you're interested in say CD45RA and CD8, if you want to just see what those look like on a 2D plot, like if you were doing manual gating, you can do that all within the plugin. And then the best feature is you can take all of these values and export them to say Excel, and now you have all of your MFI statistics for these markers as well. So question. Yes, you can do that. You'd have to do it within the FlowJo workspace. I don't think there's a way to do it on this 2D plot. But yeah, and this would definitely be a good way to get clues about where you want to start doing some of your manual gating and go back to your workspace, do all that analysis. No, okay. So sorry for the virtual audience. The question was, if you see a population of interest on the Cluster Explorer, like the one I'm gesturing to with my laser pointer, you can't do any gating on this plot within Cluster Explorer, but you can open up FlowJo and ask the same questions, basically. Okay.
I can Cluster Explorer one, but you would gate that and make the population in fresh Cluster Explorer.
I don't know if you'd be able to see it in Cluster Explorer.
I think perhaps the population.
Oh, that's a good question. Like if I did a gate within FlowJo workspace,
Yeah, you
and then
that when you gated it, it would make you know in the workspace, and then you have to like restart Cluster Explorer, and then you would every one of those clusters is available. I all know but one of those
okay, cool. All right, so so this is the last slide that I have. So, I just wanted to end giving a wrap up of what I talked about for the workflow, and also point out tools that I think are very helpful and great for your analysis, but I don't have time to talk about, otherwise I won't have time to demo. So solutions on the FlowJo Exchange. So we've gone through the workflow for high dimensional analysis, right? We're going to clean up, concatenate, dimensionally reduce, cluster, maybe compare with some manual gating, and compare between treatment conditions or tissue types. So we have all of those tools within FlowJo to help you, but I wanted to just drop a name for some of the other tools we have. So for cleanup, FlowAI and FlowClean are quite popular for that. So those are going to help remove anomalies from signal acquisition. And then we also, if you want to dig deeper, like I've identified these clusters, but what am I going to do with them now? Hyperfinder is a plugin to help you find the path of least resistance to gating that cluster so you can ultimately sort it. Maybe you want to sort it, do some functional analysis, do some single cell RNA-seq. So it's going to help you refine that gating strategy, remove any redundancies, and give you a panel, or I should say a gating strategy, to get to that population of interest. And then in terms of, digging deeper, Euclid is our quality control algorithm for clustering. So it helps you gauge the purity of your clusters as well as the heterogeneity between your clusters. So I highly recommend that as well as a QC check for your clustering. And we have some webinars on that topic. And also visualization. Obviously you're going to want to maybe make some graphs in another software, run some statistics, but we do have a Shiny plugin called Violin Blocks where you can immediately take data from the FlowJo workspace, spin it up on the web, and it'll make some nice violin plots of expression levels. So, and you can copy those images, put them into a PowerPoint, but it's just a quick way to get your data from the workspace onto some kind of a graph. Okay. So time for the demo. Are there any questions on the content while I change windows?
So hyperfinder is applied to after the dimensional analysis and you identify a cluster, then you apply the, okay.
Yep.
And then that'll show you the gating path to get to the cells that were within that cluster. Okay.
Right. Yeah. And it'll remove parameters that it finds unnecessary to just kind of help you get a refined gating strategy, and it gives you a measure basically of like the confidence of the gating. So 90% chance of capturing all of your events in this cluster with this hyperfinder gating strategy.
And if you've got two slightly different phenotypes within that cluster that have kind of clustered together like a low-high population, will it kind of break that out, or?
That I'm not sure, probably not, but once you get your gating strategy, I imagine you could refine it on your own at that point and be like, okay, well, once I get to this, I'm going to want to do low, high, or separate those into two clusters and give yourself two different gating strategies. Maybe they wouldn't be the same. Yeah. Good question. Okay, so I'm going to do just a quick demo so that I can have you all out of here in a decent amount of time. So just to save some time, I'm opened up a workspace. Oh, that's weird. What happened to my, you guys can't see that, can you? That's weird. I feel like everything's still plugged in.
Yeah, worse comes to worse.
Yeah.
It's like they're on, but it's sending black. Do
Great.
you have a quick display?
Oh, there we go. Okay. Apologies to the virtual audience. We had some trouble with the screen sharing onto the projector, but I think we are good to go now. Okay, so I've opened up a workspace, loaded up some samples. This is a pretty simple experiment. It's a roughly 30 parameter panel, unstimulated PBMC, and PBMC is stimulated with PMA anamycin. There's a bunch of T cell lineage markers in here, as well as some activation markers. And we're basically going to ask the question of what the stimulated cells look like and what new populations we have after stimulation. So I've done some cleanup down to my live T cells, which is what I'm ultimately interested in for dimensionality reduction and clustering. So first thing we want to do is go ahead and add some metadata so we'll be able to separate the two files from one another after we merge them. So we're going to go over to workspace, keywords, add keyword. I'm going to call it condition. And so for those of you that weren't here this morning, I always add an asterisk before my personalized keyword just to make it easier to find later when I have to add it to the concatenated file, which you'll see in a second. So I've added my keyword, which is basically condition. Now, when you add your keyword values to each of your samples, you're going to want to use numerical values. Otherwise, we're not going to be able to gate on it after we merge the file. So if I write unstim and stim, that's not going to be gateable in a plot window. So in this case, for the sake of concatenation, you're going to want to use numerical values. So I'm going to give my unstim a value of 1, my stim a value of 2. Okay? Now you can imagine if you had a more complicated workspace, you could add keywords for tissue type, keywords for donor type, keywords for time point. So you would do all of that prior to going to concatenation. Okay? But for the sake of simplicity, I've just got a simple keyword here for condition. So now I'm going to pick the cell type that I'm interested in doing the analysis for. So I'm going to pick T cells from each of my files, right click, export concatenate populations. And this is going to open a dialog window. head over to the concatenate tab. And this is where we're going to start merging our file. So events to include. So remember, we talked about normalization. We don't want to have more cells in one file over another. Also, for the sake of speed, I'm not going to include a whole lot of events. More events you include, longer the algorithm's going to take. Okay, so you certainly could include 100,000 events, because I have that for each of these samples. But in this case, I'm going to tell the concatenation, I only want you to grab, let's say 20,000 cells from each of these files. Okay. Then you can name the concatenated file if you want to. So maybe I'll call this concatenated T cell. Okay. And then choosing parameters, I'm going to do all uncompensated parameters. Oh, I apologize. Question back there. It's supposed to be equal distribution, so random, but yeah, should be representative of your data set. So question was, how does it pick the populations that you're trying to include? The answer to that is it should be representative of your data set. Okay, so I've named the file. I've chosen all uncompensated parameters, which you'll typically want to do, especially if you've done any compensation matrices within FlowJo. But if you did all of your compensation during acquisition and you don't think you'll be wanting to modify it later, you can do compensated parameters only. But I'm going to do all uncompensated. Concatenate all files together. Now, this is an important step here. Choose keyword. If you don't add keywords to your concatenated file, then they're not going to be there when we're trying to separate them and get them out. Okay, so why isn't it giving me, there we go. So right here where it says additional parameters, I was in the wrong Dropbox, apologies. So concatenate all files together, additional parameters. So it says no keywords selected. You want to make sure you pick, oh, sorry, I keep looking down and not up. So that's going to, and Howie can correct me if I'm wrong, going to include both the compensated and the uncompensated.
Well, I mean, it's very confusing on why.
Yeah.
The answer is because we'll apply the comp later. So it's assuming, so the question, better question is why we choose the comp. Other answer, but well, if you have variable compensation matrix applied, but if all your samples share the compensation matrix, you want the flexibility to change that compensation matrix, touch the whole matrix or value later. Use the uncompensated data. If it's like day 0 had ABC size 7 to PE 14, day 7 it was 18. Like if that varied, you want to concatenate the compensated parameters to make sure that that compensation is locked in. It needs to be for, because once you do that, it's 1 to 5. Yeah. So you can't vary the compensation.
Mm-hmm. Yes. All right. And please feel free to yell at me if I'm looking at the screen and not seeing any questions in there. Okay, good, good.
If you do uncompensated parameters, you have to then apply the compensation matrix to that concatenated file after. So FlowJo, it'll see if all of them have the same comp matrix applied beforehand, it'll automatically apply that either to the existing workspace or a new workspace. It'll do it for you. But if you have variable ones, which one is it supposed to choose? It won't. And in that specific circumstance, if you have different compensation matrix applied for specific reasons, then you probably want to do compensation matrix. But if there's just a small amount of variability, then just choose one.
Okay, so uncompensated will bring over the matrix and uncompensated parameters together.
Right.
Okay.
Yeah.
It'll act just like.
Yeah.
Mm-hmm.
And then you can tell, I'll throw in one more thing. You can tell if you chose compensated parameters because the new parameter will be called FJ comp instead of just comp. And it is locked in. You can't do anything about it. That's your thing.
Yes. Thank you, Howie. And okay, so adding your keyword. So we're going to go ahead and do choose, add the keyword that I added earlier, which was condition. So I'll scroll down. So again, this is where proceeding with an asterisk can come in handy because it helps search for them much more quickly. So I'm going to add condition, hit OK. And now we are ready to concatenate. Oh, the last step is you can change wherever the concatenated file ends up. I believe it typically tries to put it where you are from the workspace where you're working, but you can change that if it has gone rogue and put it in a different location. So I'm going to go ahead and put it in the folder where I think it should go. Okay, so once we're ready, you can hit concatenate, right? And you're gonna you can make a new workspace if you'd like. I usually just like to put it in the same workspace. Go ahead and into existing workspace, hit close. All right, and now here's my concatenated file, and you can see there's 40,000 events because it was 20,000 events from each sample, so 40,000 total. And I'm gonna go ahead and move this into another group. Okay, so now I have my concatenated file. So let's go ahead and go forward with the dimensionality reduction. I'm gonna go ahead and use t-SNE today, which is native to FlowJo as of version 10.6.3, as of version 10.3. So, but if you're on 10.8, it's definitely there. So I'm gonna go ahead and use t-SNE, which is here as a button in the activity ribbon. So I'm gonna hit t-SNE and select my parameters here. So in terms of parameters of interest, you'll want to exclude ones that aren't going to add any discriminatory power. So if you recall, I already did some gating to get down to my T cells, right? So I probably don't want to have something like live dead. I probably don't need CD3 because I already gated down to that. So you'll want to remove maybe the parameters that you've already, they're going to be homogeneous in that population, not going to give you any discrimination. And I'm going to go ahead and run this and then open the window again and talk about the tunables, but I just want it running in the background while we're talking. So go ahead and do that. Okay, so we'll have a little progress bar up here. So let's open up that window and talk about some of these tunables here. So we talked about the parameters to pick. Learning configuration, I would recommend OpsNE. It's an optimized algorithm for t-SNE. Optimized for speed and also just for better resolution. So it has automated the learning rate as well as the number of iterations to get to your final t-SNE based on the size of the data. So it really removes the need to have to deal with any of these knobs down here like iteration, perplexity, and learning rate. So it's optimized those, hence t-SNE. So I would recommend that button. And if you want to learn more about the details of that algorithm, we have a link there to the publication. And I believe Anna Belkino just gave a webinar on it through ISAC. So you can watch that if you want to hear a little bit more about it. KNN algorithm. So you have a couple options here. There's exact and approximate. As the names would imply, exact is going to be a little more exact for the dimensionality reduction, might do a little bit better at resolving rare populations. But if you just want speed and are willing to make that small sacrifice for resolution, you can go with ANNOY. So that's going to be faster. Gradient algorithm, Barnes-Hut, just the original t-SNE, FFT, or fast Fourier transform interpolation, just another boost to speed improvement. I can't say that they're dramatically different in terms of what the outcomes are, but FFT is definitely faster. So if you want to just go full speed, you'll go ANNOY, FFT. You want to be a little more exact, go grab a cup of coffee while you're running this. You can go ahead and do exact and Barnes-Hut. Yeah. Like what kind of specs on your computer. So technically all of this can be run, on 16 gigs of RAM, typical workstation. For FlowJo, RAM is power. So if you want to bump up your RAM and you have the ability to do that, certainly a good recommendation. But you can definitely run these with a typical benchtop computer. Okay, so our t-SNE is finished running. Let's take a quick look at it. So here's our dimensionally reduced space. This is on our concatenated file. So you can see we have some islands here, maybe one island out here that might be of some interest. So a couple of things you can start looking at immediately. One is, I'm going to drag this down here, make that a little bigger. There we go. So if you change your pseudo color down to a heat map statistic, you can unlock a third parameter that you can overlay and immediately see in your sample, right? So here I have sample ID. So maybe I want to ask, well, where's all of my CD69? Oh, here's some CD69 expression up here. Okay. And where's all of my FOXP3? Oh, well, I've got a nice island here, really hot for FOXP3. Maybe I have some Tregs there. Okay, so that's one way you can start fishing around on the dimensionally reduced space, maybe getting some ideas of what kind of cell types you might be interested in dissecting. Before we play around on this, the next thing I'll do is, so remember we did our keywords. And so now those are going to show up as a parameter down here. So here's condition. If I change this to histogram, right? So now I have my two different conditions here. So I have a 1 that I'd use for the unstim and a 2 that I'd use for the stim. So now I'm going to go ahead and gate on those so I can separate them out. So I've got keyword value 1, which was my unstim, and keyword value 2, which was my stim. Okay. All right, so now you can see that it's created those two populations, and this one's a little less than 20,000, probably because I need to just go in and adjust some of the gates. But you can see that they're close to 20,000 in each, so unstim and stim, which is what we grabbed during the concatenation. Okay. So if we go over to our layout editor, so there's a couple ways we can start looking at the t-SNE and dissecting what's in each of the islands. So one is you can just drag your concatenated file here, change your parameters to t-SNE1 and t-SNE2. Okay, and then let's open up this plot and do the same thing. So let's say I was doing my third color heat mapping and I became interested in aisle 22. That's a nice little hot spot of aisle 22. I want to know what's in there. Okay, let's go ahead and draw a gate on it. All right, call it whatever you want. Put a question mark. You want to be fancy, you can call it query. So now I have a query gate on that dimensionally reduced space. Oops, there we go. So if you go ahead and undo this, I'm going to drag that query gate and ask, well, how are they different within my stim and unstim condition? So you can see in my unstim, there's 3% frequency of that query gate, 3% frequency in the stim condition as well. And then if you're looking here on your t-SNE reduced space, you can also do some adjunct histograms and start asking what's inside of that query gate. So if we do something like that, let's go ahead and drag, say, for instance, my concatenated file here, drag in that query gate, right? And then if you right click it, adjunct histograms, oh, sorry, that's the wrong function, although that does look pretty crazy. Multigraph overlays, is that it? There we go. So multigraph overlays, histograms, open that up. There's probably more parameters here than you'd be interested in, but you can start looking into what's inside that query gate, right? So you can see that in there, I've got some interferon gamma that's higher relative to the rest of the dimensionally reduced space. Let's see, what else do we have? Maybe a little bit more CD39, which is also popular for Tregs. A little bit of Ki67. So you can start moving around that query gate and asking, what's inside each of those islands. Okay? Some other popular visualizations is, let's say you were going to drag in, remember we designated our stim and unstim condition. So let's go ahead and just drag those in. Unstim, stim on the right. And we'll make that, we'll go back to our t-SNE visualization. Okay. And so you can see how there's some islands that are not in my unstim, but I have some islands that show up on my stim. So these could be populations that resulted as a after the stimulation condition. So you can look at them pairwise this way, change colors on them. Maybe you want to make your unstim a different color, make it gray or something. And on your other plot here, maybe you'd want to make that red for stimulation. right? So this is just, you could do a side-by-side comparison between each of your conditions. You can also make an overlay, which we learned a little bit about this morning, right? So now I've got my unstim and stim. I can easily visualize which islands might be more attributed to one condition over the other, okay? So you could play with this all day, move this query gate around, find, well, here's my CD8 cells, here's my CD4 cells. But then ultimately we want to identify populations and find what those populations are. So let's go ahead and move on to clustering. So I just wanted to show you a little bit of how you could visualize some of this t-SNE space. I guess one last thing I'll show also before I leave the t-SNE is another popular visualization is to see other markers and where they fall within the dimensionally reduced space without having to copy and drag all of them. So if you do that, multi-graph color mapping, let's just pick a few of these. Okay. And you can see off to the side here, you see IL-21, FOXP3, TCR, gamma delta, not so much activity there, a little bit of perforin down here. And so you could pick all of your markers and see where they fall on the dimensionally reduced space if you wanted to just start getting some ideas of what these populations are. Okay, so now let's move on to the fun stuff. Let's go do some clustering. So I'm going to go ahead and pick FlowSum just because I know it's fast, but so let's go ahead and do FlowSum. Open that up again, pick the markers with some discriminatory power. So I'm going to get rid of live dead and I'm going to get rid of CD3. And for FlowSum, you do need to kind of guide the algorithm into how many clusters you might expect. I'm going to go ahead and say something like 15, because I think I have about 10 to 15 different cell types in this panel. This is something you can tune. You can always run FlowSum again and bump up the number, bump it down if you think that you've under or over clustered. All right, and then you can also within the advanced tab change the way that the minimum spanning tree is going to look. So there's a bunch of extra features on there. So let's go ahead and hit OK. Let that run in the background for a second. And I guess while that's calculating, the other thing I'll mention is where do you get all of these plugins, right? So while we're waiting for that, you go over to plugins. There's already a hot link there to take you to the FlowJo Exchange, which is where we house all of our plugins. You can filter and say, I'm not interested in SeatGeek, get rid of that. I only want to see the dimensionally reduction tools and clustering tools. And so it's just a filter to help, get to your plugin of interest faster. You click on each of the information cards, you can see a little bit more about the algorithm and you can get notified of plugin updates if you want to sign up for that. And you can also see previous versions of all of the other plugins as well. Might be some notes on compatibility, so that's something to watch out for. For instance, FlowSum, you can only run version 3 on a certain version of R and 2.9 on a certain version of R. So something to keep in mind if you're going to be installing a lot of these. So our FlowSum has finished running. So you can see it generated the populations. Whoops, I don't know what happened there. I just accidentally did a copy paste. There we go. So it generated each of the FlowSum clusters right under the concatenated file. And so one nice thing with FlowSum is it does generate a minimum spanning tree. So if you go over to your layout, drag that FlowSum node into your layout editor, you can get a nice tree to see your clusters. And then within each of these meta clusters, you'll see the more markers you have, the harder this might start to become to see. But within each of these meta clusters, there's basically a pie showing each of the markers that you chose for the clustering algorithm and their frequency, which is the height of the bar within each of these meta clusters. So it generates a nice image, also puts it in an output folder where you're working in your workspace so you can get a nice JPEG to put into a PowerPoint or something like that. So it does generate a really nice visual here. So the other thing you could do is if you had your t-SNE plot, you just want to see what these clusters look like on a t-SNE, you can go ahead and do something like that. Go ahead and overlay them. Right, and we can see now my query gate is largely this cyan blue population, so probably like cluster number 14 or something like that. So that's one way you can visualize those. And you can also, we can do this within Cluster Explorer too, but if you wanted to drag these clusters under each of your stim and unstim, you could start asking questions about their frequency in the statistics tab. But again, ultimately, I think the best tool to start figuring out what these clusters are is to use Cluster Explorer. So I'm going to go ahead and just launch that rather than, do anything too laborious with these clusters. So I'm going to go ahead and go Plugins, Cluster Explorer, pick your clustering. So if you had done multiple clustering algorithms like phenograph, FlowSum, XShift, you can pick all of them at this stage and look at them all at the same time. Again, pick whatever markers you're interested in looking at. Maybe remove the ones that you're not as interested in. And pick your dimensionality reduction algorithm. So if you used UMAP, pick UMAP. In this case, we use TISNE. I'm going to go ahead and go with TISNE. Hit OK. And so once this launches, it's going to give a bunch of different information, right? So here we go. So we have a bar chart showing the frequency of each of the clusters. We've got this chord plot showing the relative expression level of each of these markers. So these are all the markers that I picked. So you can see granzyme B positive, IL-22 positive. So everything that I put into Cluster Explorer, I can now see here. I also have a heat map of relative expression as well for each of the markers. More you have might get a little tricky to see, but I'm going to try and make it as long as possible. And you can see each of your clusters here on the left. We also have the dimensionally reduced space down here. And then here's the control panel where you can ultimately get Cluster Explorer to do a bunch of different things. So one cool thing is if you want to just sit back, play through your clusters, you can watch them, see how they're falling on the dimensionally reduced space, where they're falling on the heat map, and just start interrogating which ones might be of interest, which ones might look different. So you can play through all of your clusters. You can change the speed for that. In terms of the profile points, you can do the mean, or my personal favorite is the median, especially for logarithmic data. So you can change that. If you have a particular phenotype that you're interested in, you can start using some Boolean logic to zone in on those. So maybe I want to know which clusters have FOXP3 positive, and I want to know which ones are TNF alpha negative, or I want to know which ones are CD4 positive, right? So you can make a series of gates and it's going to start refining which of those clusters have those markers of interest. You could also do a not gate. I want something that's not positive for that marker. And now you've zoned in on a particular cluster of interest if you have something that you're digging for that you know is of interest to you. If you want to clear out anything you've done, you just double click the white space. And then on the heat map view, you can also, and this is what I think how I was alluding to earlier, you can add nodes to your heat map. And that's really going to help you visualize the frequency of these clusters between your different nodes. So here, for instance, I have a node for each of the markers. I go to nodes and then do add nodes as columns, right? So let's add my stem and unstem, right? Let me make this a little bigger. So I'm running out of screen space here, but you can see that I have my unstem here on the right and my stem here on the left, and each of my clusters. And so using this heat map scale, you can see that, for instance, in the stimulated condition, it seems like I have a lot more domination of these clusters here. So clusters 5, 6, and 7. So if you wanted to start asking what those are, well, what's cluster 5? Well, cluster 5 is CD69 positive, it's CCR7 positive, it's IL-22 positive, granzyme B somewhat intermediate. So you could start learning like, hey, this is an activated T cell, or this is a Th17 cell if you see that it's positive for IL-17, or this is a Th22 cell.
So why do you have a gradient on your stem and unstem node there?
A gradient.
Shouldn't it just be one or the other?
Oh, I see what you mean. Like, shouldn't one cluster be entirely? Yeah, so there's a gradient because a cluster, it could be attributed to just one condition, but it could be that population is in both conditions, it just became more frequent in the other.
So it's saying how many of the stem and unstem were in that particular cluster.
Yes, yeah, or their contribution, I would say, yeah. Yeah. So let's see. Yeah, so you can start teasing apart, which of these clusters might be of interest. Maybe you have a cluster where, let's just pick one, and I want to know, well, what does that IL-22 look like? Is it really negative? Is it really low? You can, within Cluster Explorer, just go in here, change the access to IL-22, which I seem to have passed. There we go. And maybe let's just say I want to see IL-22 and whatever CD8. Then you could see that IL-22 on a 2D plot as if you were in your workspace, and you start picking other clusters, you can compare them to one another. So maybe I want to compare it to those clusters. Now I can see all three of those and say, hey, this pink cluster has more IL-22 in it. So you don't even have to leave Cluster Explorer till you're ready to start doing some exploratory gating. Okay. You can also copy these values. So if I double click the white space again, copy content. Oh. What's that noise, Howie? Oh my goodness. Let's try it one more time. Okay, it was working this morning. Maybe it's a screen share issue. But you do copy content, can paste it onto Excel, you get your values there. They'll have to be delimited with some kind of a character like quotation marks or a space, but you would see those values populate here in the Excel table. Not really sure why it's not working right now, but we'll leave it as is. And
the Java FX app, and they get out of sync with the calculation engine sometimes.
okay,
Probably screen share.
yeah. So for the sake of not cursing you all with the boing of doom again, I'll just go ahead and say you can also copy these images onto a PowerPoint, onto your layout editor. So if you right click the image, you'll have an opportunity to copy to the clipboard. Yeah.
Feel like you showed that on the workspace plot, but
Oh, the 2D plot here. Yep. I just basically changed the axes. So if you recall, the default view it gave me was just the Disney, but you can come in here just like you do in a normal workspace and change these to anything you like. And you can copy these images too if you just want the pretty colors to compare the different clusters. Let's see, I think that is pretty much all I wanted to go over. So I just wanted to end with saying that, you know, ultimately you can make these comparisons between these two groups. So we've learned that maybe in my stimulation condition, I've got more CD69 expressions and more activated cells. So you can really start dissecting, you know, which of these clusters are more frequent in a condition of interest, and then using Cluster Explorer to start phenotyping what they are. You can export stats out, compare not only frequencies, but also MFIs of any markers, and you can confirm anything with some manual gating. So if you become interested in a TH22 cell, you can go back into your individual files or your merged file, start manually gating for TH22 cells and look at their frequency between the conditions. So for the sake of time, I'll go ahead and end here. If there's any questions in the chat from the virtual audience, please go ahead and type them in. Does anybody have any other questions for me? Awesome. Okay. Well, I hope this was helpful. And if you think of any questions later down the road or would like a quick redo tutorial virtually through a screen share, I'm happy to help with that. For those of you that weren't here this morning, I left some cards here up front, but I'll go ahead and show my email again, just for the virtual audience here. Come on. So I'll go ahead and leave my email up there. You can also admire my very embarrassing Florida picture. But if there are no questions, it was a pleasure to talk with you all today, and thank you so much. And don't hesitate to reach out if you need any help.